In 1999, in a collapsed rock shelter along the Lapedo Valley of central Portugal, archaeologists uncovered the skeleton of a child. The bones were small, fragile, perhaps 4 years old at time of death. The body had been buried with care, red ochre staining the soil beneath, a deliberate act of mourning in a world 28,000 years removed from ours. But the anatomy was wrong. The limbs were short and stocky, proportioned like a Neanderthal. The chin and skull were modern. For 25 years, this child sat at the center of one of the most uncomfortable questions in human evolution. Not whether we descended from Neanderthals. We didn't. But whether something far more complicated had occurred between our two species, something that left marks not only in ancient bone, but in the cells of people alive today, walking through cities, sitting in clinics, carrying prescriptions they do not fully understand. Those marks are there. And the story they tell is darker, stranger, and more medically consequential than most people have been led to believe. For much of the 20th century, the Neanderthal narrative was clean. They were a dead branch. Stocky, cold-adapted hominins who roamed Europe and Western Asia for 400,000 years, built fires, made tools, buried their dead, and then vanished around 40,000 years ago when anatomically modern humans arrived. The assumption was simple replacement. Homo sapiens entered, outcompeted, and that was that. Two species that never mixed. This was not a fringe position. It was the mainstream. The morphological gap, the heavy brow ridges, the low cranial vault, seemed too wide for interbreeding to produce viable offspring. The Neanderthal lineage was a cul-de-sac. This was taught as settled fact. For 60 years, it went essentially unchallenged. Then, in 2010, Svante Pääbo's team at the Max Planck Institute published the Neanderthal genome, and the clean story collapsed. Non-African humans carry between 1 and 2% Neanderthal DNA, not a trace amount, not a statistical artifact, a measurable, functional inheritance woven into the genomes of billions of living people. East Asians carry roughly 20% more than Europeans. The mixing was real. It was widespread. And for 10 years after Pääbo's finding, the field scrambled to understand what it meant. But knowing that we interbred answered only the first question. The ones that followed were harder. When did it happen? How long did it last? Was it one event or many? And perhaps most uncomfortably, what did we inherit, and what is it doing to us now? The answers have arrived in a cascade. 3 major studies published between 2024 and 2026 have rewritten the Neanderthal story in ways that most people outside the field have not yet absorbed. Together, they tell a narrative that is not inspiring. It is sobering. And it starts with what happened to the Neanderthals themselves, because even that story was wrong. The old model assumed a gradual, continent-wide fade. Neanderthals losing ground to modern humans, generation by generation, until none remained. Slow decline, quiet exit. It felt logical. It was also incorrect. The reason the gradual model held for so long is worth understanding, because it reveals how the field's own methods created blind spots. For decades, researchers studied Neanderthal sites individually. Each cave, each rock shelter, each assemblage of tools was analyzed on its own terms. What was the stratigraphy? What were the radiocarbon dates? What species were present? This site-by-site approach produced enormous detail, but almost no continental overview. Nobody was tracking the density of sites across Europe through time because no database existed to do it. The picture was a mosaic of close-ups with no wide shot. Additionally, earlier genetic studies relied on small sample sizes and nuclear DNA fragments that were difficult to extract from Pleistocene bone. Mitochondrial DNA, which survives longer, was available from only a handful of late Neanderthal specimens. With so few data points, the genetic homogeneity of late Neanderthals looked like noise, not signal. It took the accumulation of 59 mitochondrial genomes and the road database spanning thousands of archaeological sites before the pattern snapped into focus. In March of 2026, a team led by Cosimo Post at the Senckenberg Center for Human Evolution at the University of Tübingen published a study in the Proceedings of the National Academy of Sciences that replaced the gradual model with something far more violent. Not violent in the sense of warfare, violent in the way that climate and genetics can be violent, silent, total, and invisible until someone reads the code. Post's team sequenced mitochondrial DNA, the maternal line genetic material that survives longest in degraded bone, from 10 newly identified Neanderthal individuals recovered from caves across Belgium, France, Germany, and Serbia. They combined these with 49 previously published Neanderthal mitochondrial genomes and cross-referenced the results against ROAD, a continent-wide archaeological database tracking Neanderthal site density through deep time. Then they ran Bayesian models to date the major divergence points. The pattern that emerged was not gradual decline. It was near extinction, followed by a fragile resurrection, followed by a final collapse. Around 75,000 years ago, as the Ice Age entered a severe phase, European Neanderthal populations did not merely shrink, they imploded. The ROAD database shows inhabited sites plummeting in number across the continent, with remaining populations contracting toward one geography: southwestern France. Northern Germany went dark. Belgium went dark. The Caucasus thinned. Archaeological traces outside southwestern Europe became scarce to non-existent. The Neanderthals who survived this bottleneck were not a diverse, continent-spanning people. They were a remnant. A biological bottleneck in the strictest sense, a tiny fragment of what had existed before. Then came a period of silence. For roughly 30,000 years, the mitochondrial record shows almost no diversification. No new lineages branching, just stasis. A small population, holding on in one corner of one continent, while the rest of the world they had once inhabited sat empty of their kind. 30,000 years of genetic standstill. Around 65,000 years ago, as climate allowed movement again, a single maternal lineage began to expand outward. From southwestern France to Iberia, through Central Europe, east to the Caucasus. By the time modern humans arrived on the continent, virtually every Neanderthal from Gibraltar to the Altai Mountains carried the same mitochondrial signature. 400,000 years of accumulated diversity, erased by ice and drift in time, funneled through one narrow line. That uniformity was not resilience. It was fragility wearing the mask of recovery. Post's Bayesian models showed that between 45,000 and 42,000 years ago, Neanderthal numbers dropped sharply one final time, reaching their absolute minimum immediately before their disappearance from the fossil record. Late Neanderthals lived in bands estimated at 3 to 60 individuals. At that scale, harmful mutations accumulate. Genetic load increases. Each group becomes more vulnerable to chance events. A bad winter, a failed hunt, a disease. The species did not need a superior competitor to finish it off. It was finishing itself. This changes the story fundamentally. The Neanderthals were not conquered by Homo sapiens from a position of strength. They were a genetically impoverished population, already in freefall, when they met a species with larger groups, wider networks, and deeper genetic reserves. The encounter mattered. But it was not the cause. So what was the encounter? Because the collapse was not the end of contact between these two species. It was the context for it. And the genetic record of that contact tells a story that the field spent a decade misreading. That is the second revelation. And it might be the most unsettling. For years, geneticists had noticed something peculiar about the human X chromosome. Across non-African populations, the X is nearly empty of Neanderthal DNA. These gaps, called Neanderthal deserts, were assumed to reflect biological incompatibility. Neanderthal variants on the X were probably harmful in hybrid offspring, so natural selection scrubbed them clean over generations. Reasonable assumption. Tidy explanation. In February 2026, Alexander Platt, Daniel Harris, and Sarah Tishkoff at the University of Pennsylvania published a study in Science that turned the question inside out. Instead of only asking why the human X lost Neanderthal DNA, they asked what the Neanderthal X chromosome gained. It seems like an obvious question in hindsight. But for over a decade, the field had been so focused on the human side of the equation that nobody systematically examined the archaic side. The data existed. The 3 highest quality Neanderthal genomes, from Altai in Siberia, Chagorskaya in the same region, and Vindija in Croatia, had been published for years. The question simply had not been asked. Platt and his colleagues compared those 3 Neanderthal genomes against DNA from modern African populations, people whose ancestors never encountered Neanderthals and carry no Neanderthal ancestry. The African genomes served as a clean baseline, free of the signal they were looking for. The result was a mirror image that no one had fully anticipated. Neanderthal X chromosomes carried 62% more modern human DNA than their other chromosomes. If the 2 species' DNA were truly incompatible on the X, both sides should show depletion. Instead, the Neanderthal X was flooded with human sequences, while the human X was nearly scrubbed clean. The team modeled 4 candidate explanations. Biological incompatibility did not fit because the Neanderthal X showed enrichment, not depletion, of foreign DNA. Natural selection favoring human variants fell short because the enriched sequences clustered in non-functional regions, not near genes. Sex-biased migration could theoretically produce the pattern, but required a complex, shifting set of assumptions across time and geography. That left mate preference. Specifically, sustained pairings between Neanderthal males and modern human females. If you have been watching what this channel has covered on ancient DNA over the past several months, you know that every major study this year has complicated the picture rather than simplified it. That is not slowing down. We are breaking down the implications of the X chromosome study and 2 other major papers from the last 4 months, and there is more coming. If that is content worth your time, subscribing means you catch it as it arrives. Here is why the mating direction matters biologically. Females carry 2 X chromosomes. Males carry 1 X and 1 Y. A father passes his X only to daughters, never to sons. If Neanderthal males consistently fathered children with human women, fewer Neanderthal X chromosomes would enter the long-term human gene pool. Meanwhile, more human X chromosomes would flow into Neanderthal lineages through those same unions. Over thousands of years, the signal accumulates. The chromosomes keep a ledger. Platt has said that mate preference provides the simplest explanation. Tishkoff has stated that the team deliberately avoided speculating about consent or coercion. Stephen Churchill, a paleoanthropologist at Duke University who was not involved in the study, has suggested the pattern implies aggression. The genetics cannot resolve the question. But the signal is unmistakable. This was not random, isolated mixing. It was a sustained, directional pattern spanning millennia and geographies. A 2024 study by Liming Lee and Joshua Akey at Princeton confirmed that Neanderthals themselves carried 2.5 to 3.7% modern human DNA, evidence of gene flow from humans into Neanderthals. Their analysis identified 2 distinct waves of modern human gene flow into Neanderthal populations, the earlier one possibly beginning as far back as 250,000 years ago, long before the main out-of-Africa migration. The exchange moved both directions, over hundreds of millennia, but not equally, not symmetrically, and not randomly. So, a collapsed species, a directional mating pattern, a 7,000-year window overlapping the final decline. The question that drives the rest of this story is simple. What did that exchange leave behind? Not in caves, not in fossils, in US. A 2024 study published in Science by Leonardo Iasi, Benjamin Peter, and Priya Moorjani cataloged Neanderthal ancestry segments across more than 300 genomes, spanning 50,000 years. They pinpointed the main interbreeding window, beginning roughly 50,500 years ago and lasting about 7,000 years, overlapping precisely with the Neanderthals' terminal decline. This was not a single encounter at a single campfire. 7,000 years of gene flow means hundreds of generations of contact across shifting geographies between groups that were living side by side. Natural selection acted fast. Within about 100 generations, both positive and negative selection had already reshaped which Neanderthal segments survived in human genomes. Some variants were immediately useful: immune genes that recognized local pathogens, skin variants adapted to lower UV. Others became useful later as environments changed over millennia. The Neanderthal deserts on the human X chromosome, the very gaps Platt's team investigated, were already present in the earliest modern human genomes tested, dated to 40,000 years ago. Whatever caused those deserts, it happened rapidly, within the first few thousand years after contact. The genome was editing its inheritance in real time. But what survived is extensive, and active, and personal. John Capra's team at Vanderbilt University cross-referenced Neanderthal variants against the electronic health records of 28,000 adults of European ancestry. They found significant replicated associations between Neanderthal alleles and depression, hypercoagulation, actinic keratosis, which is precancerous skin damage from sun exposure, and neurological conditions. The depression signal was particularly striking. Neanderthal variants near circadian clock genes contributed disproportionately to the association, suggesting the link relates to how the body processes light cycles, a trait calibrated by tens of thousands of years in high-latitude Ice Age environments, where winter daylight could drop to 6 or 7 hours. A separate analysis, using UK Biobank data encompassing nearly 300,000 individuals and more than 235,000 Neanderthal origin variants, identified over 4,300 that actively influence 47 distinct genetic traits: metabolic rate, immune resistance, skin morphology, even patterns of addiction. From the Biobank Japan project, analysis of roughly 212,000 individuals found associations between population-specific Neanderthal variants and autoimmune diseases, type 2 diabetes, and prostate cancer, conditions whose genetic architecture differs between populations precisely because the Neanderthal contribution differs. Some of these inheritances were gifts. Neanderthal immune genes helped early Eurasians fight local pathogens. One variant provides measurable protection against severe COVID-19, a benefit that appeared 50,000 years after the gene first entered the human population, activated by a pathogen its original carriers could not have imagined. Neanderthal keratin variants helped modify skin and hair to cope with reduced UV exposure at northern latitudes. These were selective advantages, borrowed from a species that had spent 300,000 years calibrating to conditions that newcomers from Africa had never faced. But the environments changed. We moved indoors. We built cities. We extended lifespans by decades beyond anything Pleistocene selection pressures could have anticipated. And genes optimized for the Ice Age carry costs in the modern world. The Neanderthal blood-clotting variant that sealed wounds faster, a genuine survival advantage when a gash from a cave bear or a flint blade could kill through blood loss, now raises the risk of stroke, deep vein thrombosis, and pulmonary embolism in aging populations. Neanderthal immune alleles that once triggered rapid inflammatory responses against Ice Age pathogens are now associated with overactive immune systems and autoimmune disorders. The depression-linked circadian variants, almost certainly beneficial for surviving dark, high-latitude winters where alertness to dwindling resources mattered, correlate with mood disorders in populations that now live under artificial light in temperate cities and rarely face seasonal food scarcity. As Capra put it, there was, and still is, a lingering cost of having this admixture. Princeton geneticist Joshua Akey framed it with precision that is difficult to improve. In certain stretches of our genome, we are more Neanderthal than we are human. Not metaphorically, literally. At specific loci, the Neanderthal allele is the majority variant in modern Eurasian populations, the archaic version 1. But the picture is unfinished. And this matters because the temptation to create a neat narrative out of fragmentary evidence is exactly how the field got it wrong before. We do not know why the interbreeding was directional. Mate preference is the most parsimonious model, but it is not the only one. Some researchers, outside Tishkoff's team, have proposed meiotic drive, a process where certain genetic elements bias their own transmission during cell division, as a partial explanation for the depletion of Neanderthal DNA on the human X chromosome. Akey himself has cautioned that the X chromosome has a uniquely complicated evolutionary history and that multiple forces may be interacting in ways that remain difficult to separate. The Platt study worked from only 3 Neanderthal genomes, all female. Future discoveries of male Neanderthal nuclear DNA could complicate or refine the model significantly. We do not know whether the 75,000-year bottleneck was entirely climatic. The ROAD database shows site contraction, but that database reflects excavation history as much as population history. Caves in southwestern France have been excavated for over a century. Caves in Eastern Europe have not received the same attention. Sites that were never dug cannot appear in the data. Post's study relied on mitochondrial DNA, which tracks only the maternal line. The nuclear genome, which contains vastly more information but is harder to extract from degraded remains, may tell a different story about paternal lineages and population connectivity. Ludovic Slimak, the French paleoanthropologist at the University of Toulouse who excavated Thorin from Grotte Mandrin, has argued that the rehabilitation of Neanderthals as essentially equivalent to modern humans is itself a distortion. He contends that both the old model, which cast them as brutes, and the new model, which reframes them as sophisticated equals, are filtered through modern human assumptions. The truth, Slimak has written, may be that Neanderthals were something we lack a framework to fully comprehend, a parallel form of intelligence that operated by fundamentally different rules. And then there is Thorin, the Neanderthal found at Grotte Mandrin in France, dated to roughly 50,000 years ago, whose mitochondrial DNA belongs to an older lineage that should have been extinguished by the bottleneck tens of thousands of years earlier. Thorin lived in the heart of the very refugium that supposedly gave rise to the single dominant lineage. Yet his DNA tells a different story, that pockets of older diversity persisted, hidden from view, long after the main genetic replacement was supposed to be complete. The surprise deepened when researchers at Friedrich Alexander Universität Erlangen-Nuremberg, analyzing the Sesselfelsgrotte Neanderthal fetus from the Altmühl Valley in Germany, found that this individual, thousands of kilometers from Grotte Mandrin, also carried Thorin's older lineage. Thorsten Uthmeier, one of the researchers, noted that the Thorin lineage was apparently more wider than previously thought. 2 individuals, separated by vast distances, sharing a lineage that was supposed to be extinct. What this means is that the southwestern French refugium may have preserved more genetic diversity than the dominant single lineage model suggests. Diversity that we have only begun to find because we have only begun to look. What else survived that we have not yet sequenced? What other exceptions are sitting in museum drawers, unanalyzed? There is one detail from this research that I keep returning to, one fact that refuses to stay in the past where it belongs. The Neanderthal blood clotting variant, The one that helped our ancestors survive wounds 50,000 years ago is still active in modern European and Asian populations. Not as a relic, not as a fossil in code, as a functional allele expressed in living tissue, influencing coagulation cascades in real time. Every year, in hospitals across the world, patients present with clotting disorders, deep vein thrombosis, pulmonary embolisms, strokes with no obvious lifestyle explanation. Some fraction of that risk traces back to a genetic variant inherited from a species that no longer exists, passed to us during a 7,000-year window of interbreeding with a population that was already collapsing when the mixing began. A hematologist reviewing a blood panel in a clinic in Amsterdam or Seoul or Sao Paulo is, in a small but measurable way, still reading the last chapter of Neanderthal history. That chapter did not close 40,000 years ago in a cave in Gibraltar. It is open right now, in every genome that carries their inheritance, in every body where those ancient alleles still fire. The Neanderthals are gone, their DNA is not, and it is not finished with us.